basic concepts of the PCR. We have done with the basic concepts of the PCR. Then we have studied very detailedly that one cycle consists of what are the steps of the PCR, the one cycle, denaturation, annealing, and the extension of that. Then we are going to concentrate today onto the quantitative real-time polymerase chain reactions, like how we are going to detect the quantity of that particular antigen. So how we are going to detect it. Okay, so this is the point what we have to do it today, and we concentrate much onto this to learn and explore the more points. Then we have got about the dice which is being used to find out the quantitative analysis. That is also is an important point here, so that we need to understand. Then what are the advantages of this real-time PCR, what we call it as, and then what do you understand by the primer, which type of the primers we are going to use it. We'll also learn on to that aspect, and we'll move on to the primary design software, so that a primer can be redesigned, can be designed in advance. So whatever the genetic material I have, I can use a software to redesign that particular, what I call it as the software of the primer design. What are the softwares we are using it here? So that one also we will have a glance on that. So exactly what is we are going to learn today. So this part we have done it. Okay, this job we have done yesterday. So we were dealing with the components of the PCR yesterday, and then we have studied this one also. What do you mean by a PCR cycle? A beautiful way I explained you yesterday regarding the denaturation, how it's going to occur. Annealing, what is mean by annealing, and what do you mean by an extension of the primers? We call it as in how this primers is being synthesized, and how we'll have the DNA fragment synthesis after the addition of the primer. And we have the new DNA strands which is being synthesized from this one cycle, participates in the another cycle to extend to give four DNA molecules. The 4 will give 16. The multiplication power we have done it. Then we'll quickly go to the step 1 and step 2. This one also I told you yesterday that the reverse transcriptase. Generally, we had an idea that being having a DNA, from there only I can synthesize it. But we can synthesize the DNA by using the messenger RNA. Now, it's very important for me what the messenger RNA contains. It's nothing but the sequence of the amino acid codes which will help in the synthesis of the proteins. So this mRNA will have definitely the nucleotide strands. So by using this one part, I can have the synthesis of this particular another DNA in the PCR cycle. So that I use the specific primers. This is the point I need to understand. In the 2 steps RT-PCR, real-time PCR is nothing but I use the specific primers. I use the specific primers. Am I audible to you? My video is visible? Am I visible in the video? Okay, fine. That's great. Okay. Now all of you just watch a small animation and just show to you. Did you see this? This we call it as in primer. The primers which is supplied. Now the DNA strand. Now I have got this. This is as the one DNA strand, the another DNA strand here. These both of these DNA strands here. By using the primer 1 and the primer 2, I have got the 2 primers here. Using this primer and supplied with the DNTPs, I call it as a dinucleotide. Whatever these nucleotides we supply with the artificial one through the solutions is going to get attached. Now, what happens once the primer is going to get attached here? This primer will allow the synthesis. For example, it is attaching here. The primer is going to synthesize the sequence of the nucleotides in this direction, to give rise to 2 DNA fragments or 2 DNA segments or 2 DNA molecules. So it's a good animation, and it's one important point. It is that the maintenance of the temperature. Did you see here? The yellow color is rising and it is coming back. The temperature which is around 95 degrees after denaturation here. When I'm using the primers here, the temperature is descending down. It means that the temperature will play an important factor, an important role in the synthesis of the new nucleotide with the help of the primary probes what I am going to use it, or the primers what I am going to use it. Now, what are the things we are going to? Now, this is what I call it as an aliquot. You can see the aliquot here. This compartment I call it as an aliquot. The aliquot compartment, I have the DNA. This is an isolated DNA. And these are the primer 1 and the primer 2. Then I have a molecule called it as an Taq polymerase. What do you mean by Taq polymerase and all those things they'll teach you in a molecular biology and biochemistry. I've got the nucleotides. This is a cytosine, this is a guanosine, this is an adenine, this is the thymine. So I have an addition of these nucleotides inside that particular buffer solution what I have got it. And then these are the nucleotides you can see here. So we have got, this is a complete picture of what an aliquot contains, This particular aliquots will have all these substances, such as a template of the DNA, where which one I wanted to make the fragments of the new DNA synthesis. Then oligoid primers, I've got oligonucleotide primers. All these are oligonucleotide primers. The plenty of them is supplied. The enzyme called it as an DNA polymerase, Taq polymerase, what we call it as an DNPTs also we have got it. And then buffer solution, which will provide the magnesium ions. So this is very important point. The magnesium ions will act as a cofactor in enhancing the reactions. You could have studies in the metals, trace metals. So trace metal is a magnesium, which will enhance the reaction of the any biochemical reaction, especially in case of a molecular biology. The magnesium plays a very important role. This is what you need to understand. This point here. These are the contents of the aliquot which will be placed inside these machines. What I call it as an RT-PCR. Now, this RT-PCR, whatever it is there. So this particular RT-PCR you see here, it's a computerized machine. Now, at the bottom, now here you can see there is some point. This we call it as a slots here. There are slots which is being present. The slots will have a smaller well. The well will be like this, and the aliquot will be placed like this. So this is used as a thermocycler. This particular thermocycler, what it does that it helps in increase of temperature and the decrease of the temperature depend upon the stage of the reaction. Now, at the upper part, what you see, this upper part will have a spectrophotometer. It is assembled with the spectrophotometer so that the fluorescent dyes, what I am going to give it, the fluorescent dyes which are present in this particular part of this instrument, which is been attached with the wells here. So this particular wells, with the help of these fluorescent dyes, will detect the what I call it as a color of that particular reagent. What is the main principle of the spectrophotometer? It is the development of the color. It's the development of the color. So I have the more antigen, the more color will be developed. I have a less antigen, the less color will be developed. So this is the spectrophotometer which is assembled here, will allow to plot the graphs against the concentration of this toxic, what we call it as a fluorospectroscopic dyes. So the dyes which are present will allow us to plot this type of a graph, which tells me about the concentration of that particular antigen. So whatever the antigen I'm looking for, either I'm looking for a DNA or I'm looking for an mRNA, whatever I'm looking for, the concentration will be plotted against on the screen of the system which is been attached here. So this is how from here I can take an output. I can get an output in the form of an output will be given in the form of the printer. So the printer will tell me about what are the cycle thresholds and each and everything it's going to tell about, so that I cannot become a fool, just a positive and negative when it says that, oh, it's a positive. No, they have to give that what will be the concentration of that particular, the antigenic substances which I am searching for. Now, this is another point. Now, this is the olden thermocycler here. What I have explained to you is the new one, what we are using it now. These are the thermocyclers. This part is a thermocyclers. Even this is one also, but this is a monochrome. What I have got it a small. Now this has been attached. This has been detached. Now with the new one has been detached. This is attached with the screen also, so that I can get operated here. This is the portable one. Plenty of the hospitals will have this. The biggest labs like the Reddy's lab, and we have the Vijaya Diagnostic, the Lucid, and even we have this our Mamta Medical College also. We have got this the new one which we have procured to do this COVID test. So these are so every time there will be an invention, and then we'll have addition of one or two other parameters to analyze and detect these antigens. Now, what are the major steps? It forms notes for you, which I am going to give you after some time. So there are three major steps in the PCR. The basic stands for PCR, polymerase chain reaction. So now when we think of a polymerase chain reaction here, there are three major steps which has been involved. We call it as a denaturation. We call it as an annealing. We call it as an extension of that particular primer. That's what we have to keep in mind. Now this I have explained to you using that picture. Now, what happens? In step 1, the DNA is denatured at the temperatures from 90 to 97 degrees. So when I want the denaturation of the protein to occur, the denaturation should be done by using the temperature. When I have the temperature, I should raise the temperature. The which part of this PCR machine will help? The thermocycler. The thermocycler is going to increase the temperature to bring about a condition called it as a denaturation of that particular DNA. This is the first and foremost important point in the steps of the PCR. We call it as a polymerase chain reaction. Next point. We have got the next point. Now, so in the step 2, we have got the step 2. So in the step 2, what is the main important point of the step 2? What I mean to say here, it is nothing but the primary analyses. So analyse what I have got at these analyses, the primary analyse what I have got, these primary analyses on the DNA template sample. So the primers which are the analyses, it means that I have shown you that animated picture. Both the primers which are present here, the two primers will start from one from 5 prime end. One will start from the 3 prime end. In between this 3 prime and 5 prime, I have an attachment of these primers, and the primers will start synthesizing the new nucleotides and placing the new nucleotide. I should use a sentence, I would call it as a placing the new nucleotides at the temperature of 50 to 60 degrees. And who will take care? Once again, the thermal cycler will take care that there should be decrease in the temperature. I cannot have the attachment of the nucleotides at the temperature of 90 to 97 degrees. I need to reduce the temperature. So we have a predefined program. So whenever the primer is going to act into the solution, automatically the thermal cycler is going to reduce the temperature to 50 to 60 in order to enhance the reaction and make the reaction stable for the formation of the new DNA strand. In a step 3, which is very important point what we are going to learn here, that the extension occurs at the end of the annihilated primers. It means that the primers which is going to get attached, extension is going to attach. The extension of the primer with the help of enzyme, we call it as an Taq polymerase. So this enzyme Taq polymerase, which plays a very important role for the synthesis of that particular primers and the extension of addition of nucleotides that approximately occurs, so that I need an increase of temperature to 72 degrees here. Starting from 50 to 60, I have the primer which comes and attached. We call it as an annealing process. So at the temperature which exceeds to 72 degrees, I have the Taq polymerase which comes into an action as a Taq polymerase, and the Taq polymerase, this is the primary strand. I have got the secondary strand here. You can see that this is the secondary. This is the polymerase. The polymerase comes and binds here like this. So that the polymerase comes and the primer comes and binds here. So once the primer comes and binds here, the polymerase, we call it as a Taq polymerase, comes and binds to the primary end, and then it starts keep on synthesizing like this. This is the primer. This is the Taq polymerase. This is the primer. The priming is binding here. So Taq polymerase comes and binds here. Then it starts placing all the nucleotides in the sequence here, making the two strand of a single DNA, synthesizing the new molecule, synthesizing the new molecule of the DNA. So the DNA polymerase 1 cannot be used to elongate the fibers. So elongation of these primers. So I use a polymerase, DNA polymerase when we use to elongate the primers. One would expect because it is not stable at the high temperatures required for the PCR. So I'm not going to use the DNA polymerase 1 here. Why I'm not going to use, I use the Taq polymerase. Why I am using Taq polymerase? Because it's a high temperature of 72 degrees. The DNA polymerase 1 cannot elongate. So I am going to use the Taq polymerase. So in molecular biology and physiology, when the technique is being asked, if the examiner is asking you that why the hell I'm not using the polymerase 1, DNA polymerase 1, the answer is nothing but that this will become deactive at the higher temperatures. But the Taq polymerase, this polymerase is active at the 72 degrees of temperature also, which helps in the synthesis of the DNA strands. This is the answer what we have to provide it here. Did you get the point here what I have taught you? Am I clear, legible? Am I clear, legible and audible? Fine, that's great. Now, we have different types of PCRs as we were discussing to you. So we are going to discuss about one which is very important. We call it as an quantitative PCR. It means that I need to understand the quantity. When I want the quantity to be studied, not a qualitative. The qualitative will be telling me about presence and absence. Presence and absence. But I wanted to study the load of that particular antigen onto the body, or the load of that particular DNA onto the body, the load of that protein molecule onto the body, or load of that protein molecule into the fluid, a load of that particular DNA in the nucleotide fluid or in the tissue fluid. So when I want to read that, it's very important for me to understand the quantitative real-time polymerase chain reaction. So the quantitative analysis will provide me the load of that particular antigen. What we are going to do, quantitative real-time polymerase chain reaction is a technique which is based on the PCR. So PCR is the one base technique. It's a base technique and an important technique. Once we have the base technique, as I told you that as the time evolved, that we have an addition of the things into the PCR, what I call it as a polymerase chain reaction, which is used to amplify it's going to amplify the targeted DNA molecules. So which type of a DNA molecules I am looking for? I have the different types of the DNA molecules. The DNA molecules give me muscle protein. The DNA molecules which gives me a nerve protein. The DNA molecules which gives me a tissue of one particular gland. So which type of a DNA molecule I am looking for? So when I want to look for a type of the DNA molecule, am I looking for the SARS virus? Am I looking for the COVID-19? Or I am looking for the Middle East Respiratory Syndrome, MERS virus? I am looking for hepatitis C or hepatitis B or hepatitis A. When I want that particular targeted DNA molecule, I need to use the quantitative real-time PCR. This is a very important thing. Now we are using this quantitative the research purpose also, and also in a diagnostic purpose. Now what it does, traditionally the PCR performed in a tube. When the reaction is complete, the products of the reaction are analyzed and visualized by using gel electrophoresis. There is a technique, we call it as a gel electrophoresis. So we use this normal when they used to do first PCR, the solution used to be extracted, and we used to have the electrophoretic machine that they could have taught me in a biochemistry in techniques, that we add to the sample site where we have to apply it, and the electrophoresis will tell me. So electrophoresis is nothing but the positive and negative ions. So I can find out that which type of the DNA which is being present in traditionally, when we used to do in olden days. But quantitative PCR is not like that, or the real-time PCR, which is a new one, is not like that. That we need not go for electrophoresis. So how it differs from the old PCR when I wanted to know that once the real-time PCR forms the solution, whatever the fluid which is being formed, then I use the electrophoresis mechanism to give me any result. But nowadays it is not required, However, the real-time PCR will permits the analysis of the product. It helps the permits the analysis of the products while the reaction is actually in the progress. So I have a formation of one cycle. Now the fluorescent spectrophometer will find out the concentration of that and then plot a graph for me for one cycle. Then the second cycle is over. Then the fluorescent mechanism with the spectrophotometer, it gives me the second reaction. So that when I'm running the test, and while running the test itself, I can plot that amplification graph and concentration of that particular DNA molecule. So it means that it's an advantage for me. I need not wait till the reaction is being finished, then take the fluid, put it onto the electrophoresis, make that electrophoresis machine run on for a longer duration. It's a time-consuming process. So nowadays, what I am using that while the reaction is in a progress, cycle 1 is finished, I can plot the amplification. I can plot the amplification of the second cycle, the third cycle, the fourth cycle, the fifth cycle, and I can plot the concentration of that particular DNA molecules also. I will show you the graph, and then I'll give an explanation onto that. Now, this is what another machine. This is very important one. So once again, I have got the one-step QTR, a two-step QTR here. So it means that real time, and this is real time. Q stands for quantitative PCR techniques here, where the sequence of the primers, which is being present here, I directly add to the aliquots here. So this delicates what this is nothing but I have extracted DNA, mRNA molecule, whatever it is being present, and then the DNA polygel. This is polymerase enzyme. I'm going to use it here, and then the buffers with the DTNPs I am going to add. So this is the fluid mixture with the primers with all these things here is being added. And then in the two steps, and you see here the two steps here, I have the oligoid TCS. So these are the oligoid peptides and the dinucleotide peptide which been present. Random primers I am going to add it here. Random primers, not one. Here I am going to add the specific primers for the one-step PCR. This is one important difference between one-step PCR and two-step PCR. In two-step PCR, I am going to add the random primers, not the specific primers. Now, for example, if I am using this one-step PCR to detect the COVID, I fail to detect it because I don't have any primers for that. But I use the two-step PCR to identify this particular procedure of the new antigen which invades our body to find out its load. It is the random primers I am going to add it. And then sequence specific, this is very important for me. Sequence specific primer, random primers with the sequence specific. So I have an addition of random primers which will add with this, and then this will be added into the solution. And I add a cDNA. This is a complementary DNA particle molecule I'm going to add, and then I'll have the formation of the sequence, special sequence of this fiber, qPCR. It means that I'm going to identify the type of that particular DNA molecule, what I am looking for, or what I am looking for. When I don't have idea, when I don't have a pre-synthesized primers for that particular antigen. So the basic difference between the one-step PCR and two-step PCR is nothing but here we add the random primers also, not sequential fibers, random fibers also additionally added so that it helps in the synthesis of the new DNA. This is one important point. The one I'm going to do. So now in the real-time PCR, what are the steps of this? We need to understand the steps of the real-time quantitative PCR. It's a flow chart which tells me about how it's going to work and how much I should write in the examination. It's a bullet points, what we call it as. Point number 1, the customer is fill out the customized form for the qPCR. There will be a form which will be given to the customer, in the sense the patients or a doctor is going to fill that, what he is requesting for the qPCR, which type of the DNA molecule he wanted to be plotted in this particular PCR. And then relative information about the detect of the genes. Now, which type of the gene he wanted to detect? That one also. So it means that here the primus which is already is been designed. Say that I wanted to find out from the amniotic fluid or the fetal fluid which is being extracted, that I wanted to find out about the genes which cause an autism. So that they write that I am looking for an autism. I have already got the primers for that. It is pre-designed primers. So I throw that primers into the solution, then run for the detection whether that particular fetus who's going to born after the 9 months, once the gestational duration is been over, will suffer with or not, so that I can take a precautions, or I can take any what you call it as an a line of the treatment to avoid that particular type of genetic diseases which the fetus will be born here. So detected genes, whatever we wanted, we will write that. That's an important point we need to understand here. Second, design and synthesis of the qPCR primers for the detected genes. What we have that I'm looking for an autism gene. I'm looking for that one particular hepatitis B gene here. So that what these people will do, that the ready-made primers what they have, they throw into that particular fluid. So they prepare it. How they prepare the primers, I'll tell you by using a software. Now, DNA, RNA samples is extracted and purified. As I told you that how the DNA and RNA samples will be extracted, and then the reverse transcription of the RNA is being performed. So that we were using the reverse transcriptase enzyme. So we perform the reverse transcription. If I'm looking for an RNA, DNA from an RNA, DNA from DNA is okay. RNA from DNA I'm looking for. Reverse transcriptase polymerase enzymes. Reverse transcriptase enzyme I need to use that. Now, what it does once we do that, now perform the pre-experiments of the qPCR to test mainly the specificity and amplification and efficiency of the qPCR primers So I perform the pre-experimental types. I'm not going to throw directly the report and put the test solution and give it. First, what I do that with the little amount of the fluid which has been present, I perform the pre-experiment. Whether the primers what I am given to you is that primers we are looking for that particular DNA, helps in the synthesis of that particular DNA or not. Is the amplification it is giving me correct or not? I call it as a pilot study. Generally, we call it as a pilot study. So pre-experiment part also I am going to run it. That report I am not going to give it to our what do you call it as the patients or to the doctors. So I give it the complete. Once pre-experimental part is being correct, then I'll run the sample. Then I will run the sample. Now we are going to perform the qPCR. We call it as a quantitative PCR to detect all types of the samples. So this is what we call it as a quality analysis. So when I switch on the PCR machine, I have one term. We use a term called it as a quality control. It's a very important point for the diagnostic laboratory medicine. If that particular machine is not calibrated with the controls which is being given and supplied from that particular company, I have an increase or decrease of that particular ranges. So the quality analysis will be done. How the quality analysis will be done? That sometimes say that, for example, we have a machine called it as an auto analyzer. We have got an ELISA CLIA model, what we call it as an. So when I am running the CLIA and auto analyzer, I get some sample from one Tom, Dick, and Harry. It's X institution, not my own one, from X institution. And then what I do, I run that samples and they give me some values. I match with that. If I'm matching with that, it means that I am following the quality analysis of my machine, particularly it is being correct. So like that, what we do, we counter check with the other samples also. So before starting a machine, we are going to do this, what we call it as a pre-experimental study to find out the quality analysis, quality control board we are going to use for that. So once it's been done, perform the qPCR for the n number of the sample per day. Once is enough. Per day, I'm going to, or when the reagent kit is been over or exhausted, when I'm filling the reagent kit back, that time I'm going to perform this particular type of quality analysis. Then now analyze the experimental results. Now what I do, I analyze an experimental results later on to that experimental results, and data will be plotted and we'll present the report. So this is what we call it as a process. What is the process? The process which tell me about that, how should I run the test? Point number one. No, I generally what I do that I don't perform the point number 4 here. I am at a bias. My results may go high, my results may come down. Got the point here. Now, for example, I am synthesizing the DNA from an RNA. I'm using the reverse transcriptase enzyme. I may not get the results. You got the point here? So that I be prepared for that what I want. So what I want, definitely I want to detect the genes which is told by the customer that I am looking for that specific gene. Recently, we were doing one work onto the uterus in collaboration with the Kamneni Life Sciences in the uterus. Now, fibroids which are developed on the uterus, the cysts which are developed on this, we extracted that, and then we wanted to know that which gene is causing this, because you see the plenty of stress condition, fibroids and the cysts are formed on the females are becoming, they are not able to conceive. So we just wanted to find out which gene is causing or whether it is related to the stress. So we want to detect it. It means that we started working on to the genes which is from where the uterus is being formed. Did you get the point here? And how mutations is being occurred, you're taking the condition, call it as a stress. When the people who are exposed to the work stress or familial stress or money stress, whatever it might be, the stress is a factor. Is it bringing or not? We want to compare the genes of the both. So we were doing that particular. So it means that which gene I am looking for the point of a research, point of a diagnostics, which gene I am looking for. It is very important for me to tell that I am looking for a gene. Then I need to detect that particular primer. Primers needs to be synthesized. Got the point here? So this is how the whole flow chart for the PCR will work out. Point number 1, detection of the genes which is being detected. Then detection of the primers. I need to detect the primers and synthesize the primers. Then I have to find out that am I synthesizing a DNA from DNA or RNA from the DNA, so that I will as the reverse transcription enzyme. And then I need to confirm the quality analysis of my instrument, whether it is working properly or not. Blindly, I cannot switch on, you throw the report onto that. So I perform quality analysis, quality control test I'll run in the form of the pre-experiments. Then what I do, then I perform the qPCR perfectly to analyze, and then only I give the result onto that when I am confident that yes, my machine is correct, I don't have any bias. What happens if I throw doing the pre-experiment this one, if that report, if the person gets in doubt that no, I was fine, but this particular report detect me as a disease, he go to the another lab and get it tested. If they follow the quality control measures here, I'll be at a fault. When they put me behind the bars, making a nuisance to the patient. So I cannot compromise onto the quality control of any instrument. I should see that the quality control analysis should be done before I start that particular machine. Any machine we switch off and on, we have to do this quality analysis in laboratory medicine, which is the best growing branch in the medical field nowadays. Did you get and understand the flow chart, what I have told you? Great. Now, this also facilitate the quantitation of the DNA. When I'm speaking about the quantity, it means that qPCR. So quantitation of the DNA is made possible by using quantity. It's an advanced technique what we are going to do it. Okay, the next point is achieved by using the various fluorescent dyes. As I told you that the fluorescent dyes. The fluorescent dyes will give me the quantity of that particular DNA molecules. Yeah, this is present 100 millions, 2 millions, 3 millions, 10 millions, like that is going to. So it can be measured. It is very easy for me to measure. So what I call it as in measuring, it's going to help me the fluorescent dyes. Now the fluorescent signals in direct proportion to the number of the PCR product molecules. It means that it is directly proportional to the number of PCR product molecules. In amplified molecules, how many get it? It is directly proportional. Number of the fluorescent dyes, number of this particular amplified molecules are directly proportional, are directly 1 to 1, 2 to 2, 3 to 3, 4 to 4. So it is easily quantified by using spectrophotometer. Data collected and data collected in the exponential phase. There's a phase, there are 3 phases. I'll tell you that what do you mean by exponential phase and experimental phase and all with a graph, with the help of a graph. So data collection, see, data collected in the exponential phase of the reaction yield the quantitative information. It tells me about a quantitative information on starting quantity of the amplification of the target. So amplification, how it is been occurred, it will tell me in a phase called it as an exponential phase. Now, these graphs will tell me what it tells me about. Now you see here, this is an exponential data which I am collected here. Now this is what about the number of copies here. I had one copy with me of a DNA or mRNA molecules here. Then this copies is been increased. You see that the copies is increased. So one cycle, two cycles, it increases the number of copies here. So as been copies been increased here onto this axis, this particular, and this axis will tell me the number of the cycles. So what I need to understand that 5 to 10, now 100 to 10. So this is 15, this is 1000, this is, and now this is this to 55 cycles. So number of cycles as increases, I have the amplification onto my left side. So this amplification will tell me about the number of the DNA molecules which has been formed and what I am looking for. But at one state, it will form a plateau phase. Because the sample will, the n number is being present. When it is detected in the sample, I cannot have the linear increase of that particular. I cannot have a linear increase like this. Definitely when the sample is being finished, when the quantifying sample is being finished, it will form a plateau phase. So that when I'm describing this particular graph here, or the graph what I have shown to you in the picture or the photo what I have shown to you yesterday will also show these graphs. So we have three phases. I need to divide this into three phases. We call it as an exponential phase. We call it as a linear phase. Then we call it as a plateau phase. What the exponential phase will tell me that the amount it started recycling it, and it gives me a baseline of that particular DNA. So when I have that baseline of that particular DNA, and automatically when there is an increase, it gives you a linear. When the quantification is over, it gives me a plateau phase. So this is the basics I'm teaching you, so that you'll be able to find out and do the analysis of that particular graph which has been provided to us with this qPCR testing. Now you see here, the plateau at cycle 25. Cycle 25, we have the plateau. Cycle 25 is due to the inhibitory effect of accumulation of PCR products. So PCR products on the polymerase. It means that the polymerase which helps in the formation of the new, arranging of the new nucleotides in the fragments of the DNA. So this is an inhibitory effect. It means that it cannot keep on synthesizing it here. I have a 3' and 5' end here. Either it should reach to 3' or 5' end. It should stop the synthesis of the DNA molecule. The length of the DNA molecule is being finished here. How? It's a big mechanism, and you are going to understand that when they teach you molecular biology, especially in the protein synthesis in biochemistry. Now, this picture, what it tells me. Now, it tells me about the cycle threshold values. You do see here the cycle threshold values, and I have a threshold line which I call it as cycle threshold value, which is very important for me. CT is very important. Now, here you see here, especially with this graph, the CT somewhere is been fixed with 23.2. So this is the normal one. So the decrease of this and increase of this will tell me about the presence of that particular DNA molecule. Say that, for example, I'm looking for an antigen, call it as an COVID-19. Now, when I map the DNA molecule for this, so when I run one cycle, see that one cycle, it is having the DNA, the compound of the DNA, the virus of the COVID, I have invaded only 100. So that in the tissue fluid, it will become diluted. So for me, I need to run the more cycle to detect this 100 molecules of the COVID-19. Now, this 100, I have got 1,000 to 1 lakh. So there is a word we use, call it as LOAD, load of that particular antigen. When I have more load, it means that the fluid which is being prepared or the sample which is being taken will contain the more amount of the virus. So when I have the more amount of the virus, I run the less cycles, more amount of the virus, and run the less cycles. So the lesser cycles will detect the more amount of the virus. So I say it is inversely proportional. So what is this? The CT value is inversely proportional to that particular DNA what I am looking for. I have the more DNA molecule, more antigen in this one. The sample contains the more antigen which is being detected. How am I detecting an antigen? Because it follows the sequence of the DNA molecule arrangement. That's what I am finding out first here. So that's why I have denaturation, then synthesis of that particular molecule. And then, so it means that the DNA which is being present, not one DNA I am giving in the fluid. When I do the mortar of that particular, or when I collect the sample from the nose or from the throat, it contains the plenty of the bacteria. Got the point here? So I do that isolation of the DNA. Then the DNA will be replicated back to a formation of the DNA. And then it helps that it identifies it. What it is doing? It is doing an identification of that particular DNA which I am looking for with the helps of the primers, So I don't know which primers are acting here. So I give random primers here. So any one of the primer will act on this and it gives me a new and the sequence of the DNA molecules of the COVID-19. So that the CT value is a what is depend upon the kit what they are going to use. Which kit which is being used is 23.2. It means that it is 24. 24 less than that, we call that the person is positive. 25 about that, we call that the person is negative in this. But this report what I have shown to you of one of the patients which I got it, so that have a 35 number. 35 and less than that is nothing but a positive. 35, 36 and above that, I call it as this is a negative report. This we call it as a negative report. So how I am going to find out? In one cycle, two DNA molecules will be synthesized. The two DNA molecule will take the fluorescent dye. And the fluorescent dye will be detected with the help of the spectrophotometer. Then it gives me one curve like this, starting from like this. When it will be detected on the number of the cycles. So number of the cycles in the sense that we have a standard sample. That's why we are going to run the standard we call it as. We give a standard. The standard is very important for that. What is the standard? The standard will give me that how many cycles I have to run it. So it's a big story to explain to you in the molecular techniques. You need not worry about that. The standards which has been supplied with that particular kit will allow me to fix one particular point that, okay, at the cycles it has been lesser than this, will identify. If I have more virus, it will identify the more number. If I have a less virus, I need because it is diluted. I given an example with the 100 COVID molecules. Then I have given an example with the 1 lakh or 100,000 with that. So the dilution is a very important factor in the sample what it is being collected in the form of a buffer. Got the point here? So this is how the cycle threshold is very important for us to detect it. And what it tells about the CT value, it tells about the load of that particular virus, So I should have, where should I think that if I have a cycle number 1, cycle number 2, if cycle number 3, no. The standards which is supplied with that particular kit will give me the standard CT value. Above that, I call it as negative. Below that, I call it as positive. Why? Less number of the cycle, I have the more load of antigen. More number of the cycles I prepared it, but yet it was unable to detect. So I call it as negative. The person is not infected. Especially whenever there is a virus load, when I'm thinking that. So when I use the drugs, for example, the drugs which is hepatitis B, which is act on the liver, the virus load when it decreasing or not, prognostic part when I wanted to know, this type of experiment help us that the load of that particular virus is decreased or not. COVID positive patient is turning into the COVID negative. How do I say that? He have not shown any signs and symptoms, but when I have done the one test, it is a COVID positive. After some time, it is COVID negative. It means that this particular antigen no more available into the body. If it is present also, it is being traces. Got the point here? So this is the one important point about vaccination, that the load will be given very lesser. If the load is given more, definitely we are going to get infected. Maybe yesterday's class, if you have heard about attenuated, non-recombinant DNA molecules, all those things you could have read it. Clear? Yeah? So this is very important point. How we are going to analyze this graph? Why am I wasting my time here? It's a very important point to you to understand what is the importance of the CT value. What is the importance of CT value? Do you have any doubts on this? Do you have any doubts, guys? Am I online? Okay. Now, that's what I told you. Now, look at this point. Once again, because we have done the analysis, how we are going to do that machine is going to act. Did you see here? This phases. This is what we call it as a particular phase. Now we have different values here. The values will be put here. How much is that? Since it is 18 here, that fellow is in positive. If it is a high CT value, a lesser CT value, mildly affected, moderately affected. When it is more than that of 35, I call it as that person. More than that of 35, the person is negative. If he is 35, I call it as 34, I call it as moderately effective. Got the point here? When it is number is lesser than that, but he is affected, moderately affected, severely affected, mild affected. I use the term depend upon the load which is being identified in the CT cycles. It is CT cycles. So every time it is important for me to tell that to ask for this cycle threshold is how much. In order to find out what is the effect, is it severe, moderate, or mild? Moderate or mild. So this is how we are going to. And then I told you that the importance of visit 1, visit 2, visit 3 will help me about to understand the prognosis of that particular patient and load of that particular antigen onto our body or that particular DNA. Because since it is a COVID report we have given, but the protein formation, the new protein I am looking for, we are not going to give this type of a report. Remember. Okay, which type of the fluorescent dyes I am going to use it? There is a dye called as in cyber green. There is a dye called as in cyber green. These are the fluorescent dyes. Fluorescent spectrophotometric methods, immunochemistries is ruling the world to identify the diseases now. Then what it contains, then I use an EVA blue, EVA green, then we have an Evans blue dye, which we use to find out the cardiac output that you are going to identify. You learn it when you come to the cardiac output in cardiovascular system. Then I have the fluorophore containing DNA probes. So nowadays what I'm giving it, I'm not going to give the dye directly to the solutions. What I'm doing is a DNA probe, what I am using. Directly I'm immersing the DNA probe into the dye and throwing into that. So such as a TACMAN, I use what is a company made, it is been TACMAN. I use a dye. It's a company name called it as a TACMAN. What is this TACMAN? TACMAN is nothing but fluorophore containing. It's a fluorophore containing, it's a fluorescent dye which is being immersed with the DNA probe. So this gives me an amplified reactions that I can have a quantification of that particular molecules which I am looking for. Now this is reverse transcriptase. This I have explained to you very well that from the mRNA I am going to synthesize the DNA. So that I have a confidence that DNA will synthesize the DNA. Now with the mRNA molecule also, RNA molecules also, I can synthesize the DNA very easily. This one also I have to have explained to you. So why I'm using, because this thermal stable reverse transcription, reverse transcriptase enzyme, it is a thermally stable. This is about the mechanism. I have presented you only the basic, a lot to explain to this about how this chromophores will act, TACMAN will act, all those things that is not required for you guys. But keep in mind that the basics are very important for us. What are the applications? This is a very important aspect what we have read. How do I use this? I use in the forensic medicine, wherever there are the crime scene or the crime I wanted to find out. Then in the diagnostic medicine, I'm going to use it plenty of times. Diagnostic medicine. So it is play a very important role in diagnostic medicine. What are the advantage of this real-time PCR? Ability to monitor and progress of the PCR reaction as it occurs in real time. I can find out like that. So diagnostic and prognostic importance in that. Then ability to precisely measure the amount of application of each cycle, how much in each cycle it contains, which allows highly accurate quantification amount so that I can give or deliver the drug with the dose. So when I identify the quantity and the load of that particular virus, I am concentrating on the drug delivery system, how much quantity of the drug I should give it. First he is infected with 100,000. Now the load is 50,000. How much drug I should give it to him? I can't give the same drug because the load is being decreased. For the modification treatment line, change of the treatment line, I am going to use of this. A very important aspect of real-time PCR. Amplification and detection occurs in the single tube. The whole tube of an liquid where the well will be there, a small slot where we are going to place it, occurs everything there, eliminating the post manipulations. It means that it's one of, we call it as an errors also will be there, that you need not worry about that. So it helps in the amplification and detection in the single tube. I not use a two tubes. I did not use the two tubes here. So real-time PCR, what we are doing, old PCR, we used to take that fluid and put it into the electrophoretic gel and then find out which type of the DNA molecule or a protein is being present. But here, only one tube I insert it, I get the result onto that. So it saves me a lot of time. And one compact machine will give me about each and every description of the particular gene. So gene analysis, gene expression analysis, I am going to use this. Then cancer research, which type of a cancer I am working for, small cell carcinoma I am looking for, squamous cell carcinoma I am looking for, which type of a carcinoma, so that the genetic relation is there or not, which mutated gene is causing the ovarian cancer, uterine cancer, fibroid or cystic. So what we are working on the project I told you, I have discussed onto the cancer research, which is related to the, what do you call it as an not conceiving the females, stress related problems. Then disease and diagnosis and its management. How do I manage it? So I can give the same drug what I have given, the load is more, the load is less, I need to decrease the dose of that particular drug. Management of that particular disease, it's going to help me in diagnosis and management and prognosis. Food testing. Definitely it is important point for us. Food testing also. Then animal and plant breeding. Why the food testing? We eat, we go to the market and buy the Lay's chips and blindly eat the Lay's chips. But the preservatives which has been added, it enters into my body and it is interfering with my genes. It bringing about the mutation of my genes. So when I want this food to be tested before coming into the market, it should be tested that is it causing any mutation in my genes. That's very important for me. Animal and plant breeding. Animals I want and plant breeding I want. Definitely I want this process to be find out like how I want different types, the crop which is grown in 1 year, I want in 3 months. Definitely I need to I do some modifications in the genetics. So this is primers, how we are going to synthesize the primers. I'll just tell you the softwares here. So it is in general, what is it mean? Keep in mind that there are 18 to 24 nucleotides in the length. That's all. 18 to 24. It curves and attaches. I have shown that animation to you how it is attaching onto the single strand of that particular nucleotide. So then we have got the primers. These are the primary bases. So these primary bases, 50% should have nucleotides of GC content. Remember this point. So 18 to 24 nucleotides will be there. So say that in 1 ml, I am giving the nucleotides, supplementing the nucleotides. I should have 50% of the GC should be there. Means guanosine and cytosine should be there as a nucleotide primers in the nucleotide primers. Now, what are the design softwares? We have plenty of the design softwares. We have a very important software called as an Vector NTI. You can see here the Vector NTI. So this is the software. What I do, a small chip will be there. I'll add that fluid, one drop of the fluid will be added and insert into the slot, which we call how we have a SIM slot. The same SIM slot, I will have a slot. The slot I'll insert inside to the machine. And then what it gives me analysis of this. So it means that, did you see this picture? So which software I'm using to form the primer is nothing but the Vector NTI. See here, Vector NTI, I'm going to use it. So I'm synthesizing the primers, because primers are the one which helps me in the synthesis of the new strands here. Don't you think it is some advantage? This is called artificial intelligence. How am I using artificial intelligence? I'm using this artificial intelligence in diagnosis. So that this particular, so I have the option to select here. Once again, you need to get trained with the software. That's a different issue. Now you see here 901. What are this? A, D, G, C, G, C, D, A, D, A. It means that the sequence of the primers, what I want, I can select it from here so that I will have that particular nucleotide synthesized and thrown into the fluid to have the formation of, because I need to add, test solution is added with the primers, So this is one software, and we have got the another software, the primer synthesizing software for PCR primer design. So I can design my own one. See that I vaguely, I think that I wanted the mutation is altered the gene. I don't have any idea. Blindly I can give. So when I type A from here, say for example, if I type A adenosine will be added. Guanosine will be added. But I need the molecular biology understanding everything. Then only I can form this. So these are the specialized people. We call it as in biotechnology people. Medical biotechnology is an evolving subject in the medical field nowadays. So we can concentrate much to do here. So keep in mind, the software will give me a designs of that particular primers. So we try to finish this. This is about the softwares, the components, the primers, and the probes, whatever it is there. This is a very important point. The knowledge is a type. What we have learned from one is the knowledge. Knowledge about the PCR, knowledge about its diagnostic important, knowledge about the prognostic important, knowledge about how the primers are being formed, knowledge about the softwares which is being used, knowledge about the COVID, how should I read the report from past 2 hours from yesterday and today. We have earned knowledge. Knowledge is a time because I have spent the time. Knowledge is a power. Now it's a power in me to read a COVID report or any report which has come from the PCR very easily. Knowledge is money. If I get trained in the PCR, I can earn money. Definitely. So the knowledge is the one which gives me a lot of events of my life, time, the power it gives me, and the money which gives me. The power when you get it when you are first. Money when you get it when you are trained in that specialization. So what are you giving here? It's the time you are giving for the knowledge. So very important platform that we are not wasting your time, that we have this softwares. Without this softwares wouldn't have been possible to for an online teaching. And then depends what we use the stylus to make the explanation on these things. So keep in mind, the time is precious. Don't waste your time. You are in the house, you are blessed, you are sitting and enjoying and learning, and you have got a good teachers. So try to understand, ask some questions. If you have, you can ask individually also and a group also, so that we have finished the technique, call it as a PCR, and we have some other techniques also. I'll try to explain you in a better way, giving some examples. There is a small video which I wanted to play to you, but it takes 1, 2 to 3 minutes. Let me see that if I can share that to you directly with web browser. Yes, I have that video with me. శ్రీనివాస్ కొంచెం ఒకసారి రా ఇలా షేరింగ్ లో మనకి షేర్ మల్టీమీడియా అని పెట్టాలా, స్క్రీన్ చూపించట్లేదు. షేర్ వెబ్ బ్రౌజర్. Guys, have some little patience. We'll try to give you a good video so that you will understand in 3 minutes. కొంచెం వెయిట్ చేయమని ఫోన్ చేస్తే కింద అయిపోతుంది టెక్నిక్. ఇది స్క్రీన్ పైన మనం పైన కావాలి కదా. ఓకే ఓకే, దీన్ని దించు, ఇక్కడ ఉంటుంది ఆర్టికల్స్ కింద పెట్టా. స్క్రీన్ చిన్న చేద్దాం, లేకపోతే చిన్న స్క్రీన్ చేయరా. దీన్ని చిన్న చేసుకురా కానీ స్క్రీన్ ఇది షేర్ అవుతుంది. ఇది ఆర్టికల్ షేర్ అవుతుంది. ఇది ఎస్ ఇక్కడ నుంచి ప్లేస్ ప్లే చేయండి. Okay, thank you so much. You are able to hear? You are able to hear? No, okay. So now this is a real-time PCR. I'm going to explain and repeat the things here. We have a 5' end and the 3' end here. You can see here. So we have got the target DNA. This we call it as a target DNA. Which segment I want to amplify in? I am going to use the primers here. The primers you can see. And we have got the DNA polymerase, which we call it as a tag polymerase. Okay, here. So this we call it as a probe. So we are going to use the probe. It will have a 2 ends. We call it as a reporter end, this one. Then we call it as a quencher. The quencher is having dye. This is the reporter dye. 3' end will have the quencher dry in the probe. Fluorescent emission transfer. Whenever it is being hit by the light, this will come and bind it here. You see here. So that so once the denaturation is been over, when I have the formation of the 2 fragments here, the probe which I am giving you with these two, the reporter and the quencher, the primers which is being present here, this is the polymerase enzyme which comes and binds. And you see here the nucleotides which I have supplied here, it's going to get bind with that particular. So now when the reporter dye, the light hits onto that and the quencher dye. So whenever it is being separated, the formation of the new DNA will be stopped. So for the next set, I have the two molecules of the DNA. Did you see here? The reaction, this one cycle is completed, the second cycle is starting here. The same thing, the polymerized story repeats here. This is cycle 2. You can see here on the right hand side, this is cycle 2. At 72 degrees of temperature also is being mentioned at this point, 72 degrees. Now the polymerase finishes its job. So after each cycle, the intensity of the fluorescent with the help of the spectrum photometer will be measured and then we plot against like this. So this is the standard one, what we call it as the black one. And then these are the colored one which is being shown here are an experimental what we have used it. An experimental. So this is how we are going to use this RT-PCR technique. In a simple way, this animation has been shown. You can go to the, what do you call it as this browsers of your browsers, and then you can watch this, a beautiful way he has explained. It's a simple mechanism. If you understand the one side, finding one, what you say, test one. So it is easy for me to find out if I have one particular DNA primer with me. COVID-19, I don't have a DNA primer, but now we are synthesizing the DNA primers. Clear here. And then we are going to have to find out the DNA molecule because we know it's a virus. I just open the, break the envelope of that virus, that mRNA from the mRNA, you know, RT-PCR, I call it as a reverse transcription PCR. I can synthesize a DNA. So the DNA will tell me about which type of nucleotide it is having. And then the fluorescent which is been taken in. One virus will take one, another one will take another one, third one, the fourth one, the fifth one. The load will take means number of molecules of this particular DNA molecules synthesized from the mRNA of that particular antigen will give me the load. So I can do the quantification. Then I say the person is affected or not affected. Hope that you have understood here. So try to see this type of material from your browsers, which will try to help you guys very nicely. Thank you so much for listening. And we will have another technique. We'll start from Wednesday. I'll take the technique once again. Those techniques I'll inform you. Okay, have a good day and same day. Take care.